This is Dr. Launen from Keene State College, and this is a short video lecture on Mendelian genetics, focusing on who Gregor Mendel was, Mendels Laws, and simple genetic systems where we track genes that occur on different chromosomes and where the genes have two alleles and the alleles exhibit what is called complete or full dominance. The picture here is a pea, Pisum sativum, Pisum, sativum is the Latin name for that. And I'm showing you this pea because it's the model organism that Gregor Mendel used. So we'll talk about Gregor Mendel, why it was important, his model organism, the laws that he came up with, the sorts of experiments that he did to come up with those laws, and that includes a lot of terminology. Ultimately, we'll also look at Punnett squares and talk about probability. So Gregor Mendel was an Augustinian monk. He lived from 1822 to 1884, and he was quite the natural historian and mathematician. He's considered by some to be the father of genetics, and what I think is amazing about Gregor Mendel is that essentially he demonstrated how meiosis works without ever actually having looked at a chromosome or understanding the chromosome theory of inheritance. He worked with the pea for a variety of reasons. One was that it is agriculturally relevant. It is a food source, and so at the monastery that he lived at, they had greenhouses that had agriculturally relevant organisms like peas. So it's a very practical organism. It also grows easily and quickly in weeks, produces large numbers of offspring. So each seed that you look at in the window there, each seed within the pea is a unique single offspring. It's relatively inexpensive. It is also capable of something called selfing, in that each plant can make both male and separately female flowers, producing male and female gametes, and therefore self-fertilizing. So you can see that makes it very handy for genetic study. It's also relatively simple to manipulate the crosses. It's a lot of work, but it's relatively simple in that you can purposefully take the pollen from one plant and use it to fertilize another separate plant with care. And this image depicts such a controlled or specific mating, where here in the top part of the figure, the scissors are being used to remove the stamens from this purple flower, preventing it from fertilizing itself. And then you can actually take the sperm-bearing pollen from another plant that you have chosen and transfer it over to the carpel or the female organs within one of these purple plants. And then you can see the results of that offspring. Mendel studied what are called binary or dichotomous traits. These are either-or traits, where there are two phenotypes that are distinct one from another. So it's not a trait like height, which is continuous, where you can measure a variety of different possibilities. Instead, it is either green or yellow. And the 7 traits that Mendel studied over the many years that he did his research are shown here. They were seed color or seed coat color, the seed itself and its color and the shape of the seed, the color of pods, the shape of pods, the height of the stem, tall versus short, and the flower position. These are axial flowers that are on the side of the plant, and these are terminal flowers that are at the end. These are all botanical terms that are worth remembering. Mendel's genius was in his approach. So he was one of the first people to really systematically conduct what we now refer to as being hypothesis-driven research and applying the scientific method as we now know it. His approach included the choice of an excellent model organism, which I've already discussed, the use of controlled crosses, and the fact that he took the time to begin with what are called pure or true breeding strains. These are also referred to as parental strains in Mendelian genetics, and we know them as being homozygous for the gene of interest or the genes of interest. He chose dichotomous or binary traits, which I've just described. And perhaps most importantly, he was quite a mathematician and he quantified his results very systematically. He used replicate, reciprocal, and test crosses. And I'll get to the words reciprocal and test crosses in a moment. Replicate means to repeat the same thing over and over and compare the results. He also used very large sample sizes and a lot of replication in all of his experiments. And this was really, we take this kind of science for granted now, but really Mendel was one of the first people to really, apply all these characteristics to genetic research. So stop for a minute. Do you know what the word dichotomous means and how that applies in Mendel's P's? And do you also remember what the word dominance means and how that applies to the concept of dichotomous traits? I want you to think about that a little bit more. And can you spell the Latin name for peas, garden peas? Mendel came up with 3 important laws. This is the law of segregation or separation, and one way of phrasing it is shown here in red. The 2 members of a gene pair segregate or separate from each other during the formation of gametes, and this happens during meiosis. Each has an equal probability of inclusion in the gamete, and progeny are produced after random combination of gametes. In other words, meiosis produces haploid gametes. Haploid gametes unite to go to diploid new individuals. He proved this law or developed it with data from what are called monohybrid crosses. Mendel came up with the concept or law of dominance. We could describe that here as the 2 members of a gene pair. That 2 members of a gene pair can differ such that one member shows its phenotype in the, that should say, heterozygote, and the other is recessed. And he proved this with monohybrid crosses as well. His 3rd law was the principle of independent assortment. And this states that during gamete formation, segregating pairs of genes, he referred to them, however, as unit factors because the word gene wasn't yet derived back in the early 1800s, that these assort independently of each other. And we know now that this is because of the random nature at which the sister chromatids or homologs align on the metaphase plate during meiosis 1. His data to demonstrate independent assortment was obtained using dihybrid crosses, which involved 2 genes. Mendel's first experiments were monohybrid crosses. This means 1 gene, mono means 1, and hybrid means you're crossing different organisms that each have the different allele you're tracking. So you're mating true breeding parental plants that show the opposing phenotype for the trait of interest. One trait of interest controlled by 1 gene is mono. For example, you cross a tall parent with a dwarf parent, or you cross a parent that makes only wrinkly seeds with one that makes only smooth seeds, or only yellow seeds with one that produces only green seeds. So you might draw this using symbols by writing big A, and that's the genotype of parent 1, is crossed with little a, and that's the genotype of parent 2. Parent 1, the big A organism, would show the dominant phenotype, and parent 2, the little a organism, would show the recessive phenotype. They're both homozygous, and that means true breeding in that they can only produce offspring when mated with themselves that look just like themselves. So here we've got one of Mendels crosses. So he takes true breeding parental organisms that are only able to produce round type peas, and they are big R, genotype, phenotype, genotype. And he mates them with another parent, also true breeding, that can only produce wrinkled shaped seeds, little r. He crosses these 2 together, and he sees the next generation, which is called the first filial or F1 generation. Every single offspring in that generation shows the phenotype of being round. Every single one will also have the genotype wherein it is a heterozygote, big R, little r. And that's because this parent can only produce gametes that have big R. This parent can only produce gametes that have little r. So every single offspring that they have will be the heterozygote. Mendel then takes these plants, lets them grow up, lets them go through meiosis, and they mate with themselves. So you get big R, little r, mating with another big R, little r, and that's selfing in the plant world. And what he sees in the next generation, which is the second filial generation, or F2, he sees that of all the seeds that he looks at in those offspring, 3/4 of them are round and 1/4 of them is wrinkled. And if you were to draw the Punnett square for this, it would be a 4x4 Punnett square. On each axis, there would be a gamete big R and a gamete little r. When those come together, you would see 4 blocks filled in, big R, 2 of the blocks would be the heterozygote, and 1 of the blocks would be little r, and that gives rise to the 1/4 or 25% recessive phenotype. So he sees this over and over and over again. So he does this, look at these giant numbers here, right? He does a lot of different experiments, and he does these experiments not just with the smooth and wrinkly trait that he was studying, but with all 7 of the traits that I showed you a moment ago. Point being that he repeatedly sees the same patterns in the F1 and then the F2. And here's some of that data taken from Mendel's original work. You can get a feeling for, just how much work this really was. He also does what is called the reciprocal cross. And I'll get to that a little bit later. He sees the same results in the reciprocal cross. So here we're seeing the parental generation with a different phenotype. Here it's flower color, purple versus white. The F1 are all purple, and that shows Mendel that the purple phenotype is the dominant phenotype. And then if you look at the F2, you see 3/4 purple and 1/4 white. So the recessive phenotype disappears in the F1 and reappears in the F2. And he sees this pattern over and over again. He comes up with the idea of an additional type of experiment, which is called the test cross. So in his test cross, he takes plants and he mates them with what is called a tester strain. And the tester is always homozygous and recessive for the trait and therefore gene of interest. So it's shown here, little p, little p, little p, little p. This is big P, and it's showing the dominant big P associated flower color. And he's showing here that he's asking the question, is this organism this genotype, big P, or big P, little p? Because the purple doesn't tell you whether it's this genotype or this genotype. It could be either. And he's hypothesizing from his crosses that this is the genotype, but he wants to be really sure. So he takes this organism and he mates it against this organism. This big P would only be able to produce gametes that were big P. This little p would only be able to produce gametes that are little p. Every organism that results when these gametes get together in a fertilization event would be heterozygous, and so they would all be purple. Purple being the dominant phenotype here. And that would show you what this was. It would show you the big P genotype. If instead this purple flower had big P, little p as its genotype, then when you did the test cross and you mated it against this, you'd have these 2 possible gametes. You'd still have the same tester gametes. And what you would see is something different in the offspring. You would see that half the offspring showed the dominant purple color and half the offspring showed the recessive white color. And that would be evidence to support big P, little p as the genotype associated with this flower. This is an idea called a test cross, which Mendel came up with. So he used test crosses to verify predicted genotypes. And I've already introduced those terms and those ideas in the previous slide, but here's a little bit more information for you if you're unsure. Reciprocal crosses, which as I mentioned earlier, he also did as part of his methodology, means that you repeat a cross where you switch the sex of the parent with regards to the phenotype and thus genotype of interest. For example, if in the first cross you start with these as the parental organisms, and big R is the male, and little r is the female, then in the reciprocal cross, you flip the sexes associated with those genotypes. So you would make big R the female, and little r the male. And that's an approach that Mendel thought of. This kind of data, this monohybrid data, showed Mendel that gene pairs segregate during meiosis. And that's what this is modeling here. Here's the genotype. Here are the gene pairs segregating to produce haploid gametes. He also used this data to show the law of dominance. And that is revealed by the fact that the heterozygote, big P, little p, is showing one consistent dominant phenotype, the purple flowers in this example. In the next video, we'll talk about how Mendel got evidence to support the law of independent assortment.